So I'm going to talk to you about my work so far investigating non-typeable Haemophilus influenzae biofilm formation in a cystic fibrosis lung environment. So cystic fibrosis is a genetic disorder in which a defect in the CFTR protein results in water being drawn out of the mucus, leading to viscous mucus accumulation which harbours bacterial colonisation. Disease primarily manifests within the lungs, but chronic colonisation with multiple pathogens results in the premature death of people with cystic fibrosis. I investigated Haemophilus influenzae, an early CF lung pathogen, which peaks in children ages 2 to 5 years before dropping off in later years. Interestingly, where Haemophilus begins to drop off is where Pseudomonas aeruginosa increases, which is a more severe late-stage pathogen. So is Haemophilus influenzae potentially paving the way for Pseudomonas aeruginosa infection? Also, whether Haemophilus forms biofilms which are more resistant to antibiotics is relatively unknown. In the lab, we have synthetic CF sputum media, or SFM1, which is made up of the average amino acids, ions, and lactate found in CF sputum. This has been built on further, containing all the components of SFM1, but with the addition of extra lipids, glucose, eDNA, and sialic acid to make SFM2. I wanted to test whether this version of SFM2, or supplemented with fetal bovine serum or nadenhemin, would allow better biofilm formation of Haemophilus. So I used crystal violet, which stains the biofilm purple, allowing you to quantify their biomass. Over 48 hours, I found that the addition of fetal bovine serum to SFM2 significantly reduced biofilm formation and instead promoted planktonic growth, suggesting this is not a suitable supplement. I then used an extracellular matrix fluorescent marker, which binds to the extracellular matrix of bacteria to biofilm and fluoresces only when bound. Using this fluorescent marker over 48 hours, I found that the addition of nadenhemin produced greater fluorescence indicative of more biofilm formation. So I concluded that SFM2 with nadenhemin was the best media for biofilm formation with Haemophilus influenzae. I then want to take this into the pig lungs, which is an established model for respiratory infection. We take pig lungs from the local butchers, cut the lungs up into squares, and infect with your bacteria of choice. This is then incubated with the SCFM to replicate the structural and nutritional environment of the CF lung. However, Haemophilus influenzae looks very similar to commensal bacteria already present on the lungs. So the model had to be modified. So I took the lungs and homogenized them with an SCFM2 and then filter sterilized to remove commensal bacteria to make lung homogenate media. Then, a simple growth curve with my lung homogenate media found that there was a significant increase in both carrying capacity and growth rate just with the presence of lung compared to SFM2 alone with my Haemophilus influenzae. So in conclusion, Haemophilus influenzae grows as a biofilm in vitro with SFM2 supplemented with nadenhemin, but the presence of lung tissue promotes growth even further. But further optimization of the ex vivo pig lung model is required before I can then investigate interactions of Haemophilus with other CF pathogens. Thank you for listening.